Okay, thank you, Simon, for being here, and a pleasure. The lecture is not by me, it's by Dr. David Blokh and me, and I must say from the outset that the credit for the whole bioinformatic part goes to Dr. David Blokh, and I just helped in the interpretation in the biological interpretation of the results. The topic is biomarkers of aging and the importance of this subject. That's a scheme. It doesn't. It's not completely legible. It's just to show the complexity of the problem. The importance of okay, I'll switch to using. The importance of biomarkers of aging is quite clear, not only on the scientific level to understand what aging is actually about, what parameters change with aging, what changes are positive, what changes are derivative, what changes are mere correlative, but also for some practical purposes, because without a reliable set of biomarkers of aging, there is little chance to test anti-aging or life-extending therapies. It is difficult to wait for many years until results are proven in real life. But with biomarkers of aging, the hope has always been that some anti-aging interventions can be tested and the results will be available quicker. Yet, despite many efforts to study biomarkers of aging, for example, by the massive American initiative on biomarkers of aging, which ended in 2000, and since then it is very difficult to get funding for this kind of work. The actual practical and therapeutic or diagnostic value of biomarkers has been rather low, and there can be great many causes for that. For example, one of the reasons could be, once again, the great complexity of the problem. Almost anything changes with age. Almost anything can be a biomarker of aging. Also, there is problem of interrelation of components. If you try to see the changes in one element, you should also know the concomitant changes with all the others. And if you try to enhance a certain element, you also have to look at all the surrounding ones in the chain. Also, the practically used biomarkers, such as those used by the European Healthy Aging Initiative, are mainly qualitative, such as if a person feels young at heart or if he's able to tie the shoes. But of course, a more elaborate and reliable system of biomarkers is needed. And another possible reason why the biomarkers hasn't yielded the results that have been expected from them is that usually the biomarkers consider correlation with linear correlations, just simple statistical measures. As something changes with age, let's take an average of a large population. And this is quite a flawed approach, because in many cases the relations in biological systems are non-linear. And that's why probably new methods are needed, such as, for example, the use of information theory that we tried as a pilot on a very limited sample data. And the advantage of information theory is it actually allows to see nonlinear correlation between parameters, unlike statistical measures. So this is once again a pilot. Nothing conclusive can be inferred from it. Just a very simple database that can be found online of about Pima Indians. It's an Indian tribe in Arizona. Quite a normal population in terms of their diagnostic parameters. Still, it is a well-recognized database on which many bioinformaticians are trained, and so we trained on this as well. We took several parameters just commonly associated with diabetes, such as insulin level and glucose and body mass index, and correlated them with age. And the purpose was to see the level of heterogeneity in different age groups. The particular feature of information theory is that it uses such measures of correlation as mutual information, and And entropy, so here we used entropy as level of heterogeneity. Simply put, it is just the number of states the system can be found in, and the more the heterogeneity, the more the variability, the greater the entropy. If the entropy is 1, it's the highest. If it is 0, there's just complete similarity between the subjects. And if the value is 1, there's just nothing common between the elements. Very rapidly, when we looked at the measure of entropy, the measure of variability or heterogeneity, we found a very interesting thing. The main result is that that in young and healthy individuals, the level of heterogeneity was quite high. That is, the level of entropy was quite high, and in older and diseased individuals, the levels of variability or heterogeneity decreased. So that is the main finding. And most interesting thing is that healthy and diseased subjects exhibited lower level of entropy, similarly to healthy and old individuals. That says that aging can be considered as a kind of a disease-like process, or perhaps a disease can be seen as a biomarker of aging. Of course, it is too early to make any such kind of sweeping statement based on such small materials, but at least it can be directionally, can be further tested on additional samples. So that's the main result: the decrease in entropy the increase in entropy in old and diseased individuals. Now to the interpretation parts. Entropy has been used to describe or to examine aging before, and the interpretations vary very widely. For example, there's the intuitive interpretation that with aging, the level of chaos, the level of entropy should increase. And that's actually the quotation from Hayflick, that there is a belief that age changes are characterized by increasing entropy. But that's quite an intuitive belief, and in order to see the actual changes in entropy, we have to consider particular systems and particular measurements that are being done. There are also some physicalist interpretations. For example, the level of entropy was found to increase in aged individuals in their brainwaves, and this was interpreted as an increase in alertness promoted state, so there's a well-known sleeplessness of the aged. There's also been interpretations of entropy in the studies of body balance. It changes the shift in the center of balance, and the interpretations once again ranges from to complete opposites. There is the interpretation that increased entropy shows better non-volitional control, which is better in young and healthy individuals with good balance. And there is the completely opposite interpretation that increase in entropy shows increased chaotic excursions that are prevalent in old and diseased individuals with bad balance. So somehow this should be combined. And also decrease in entropy has been associated with repair, with synchronicity, as for example found in sleep. Yet the dominant interpretation of entropy of decreased entropy with age and disease belongs to Goldberger's group, and he claims that with age the level of regularity increases, so that the healthy state is actually more irregular, may appear more chaotic, and this is interpreted as a loss of complexity in aging. So that's the dominant interpretation of entropy changes with aging, and that actually confirms with our findings that indeed show that with age the level of heterogeneity decreases, the level of variability decreases, the ability to change, to adapt to new environmental challenges also correspondingly decreases. this may not necessarily be such a bad thing, but perhaps even an adaptive mechanism, for example, high levels of entropy could be detrimental for the aged organism, and low levels would rather allow him to find, as you say, to find safety in a confined space, just to like a turtle to hide his head into his shell. So the decrease of entropy may be a protective mechanism. Still, nonetheless, young and healthy individuals are characterized by high levels of entropy, high levels of heterogeneity and variability, and this can actually serve as an indicator of some anti-aging interventions about seeing their ability to restore the level of entropy of heterogeneity. But once again, it's too early to make any sweeping statements. Much more data will be needed, and we are looking for data, in particular clinical data in relation to age, not just any, but data that can be easily tabulated or already tabulated. So I'll be happy to speak about this. There may be additional applications of information theory, once again using measures such as normalized mutual formations and entropy. And these measures are more beneficial as compared to statistical methods. Once again, they allow to see the nonlinear relations. Secondly, they allow to see combinations of several parameters. For example, it is possible, and we've done some preliminary study not shown here, hopefully someday. For example, if you take a single parameter and correlate it with a certain disease, it shows almost no correlation. But combining several parameters with age, you can show quite a good correlation. So age can be seen as a kind of a meta parameter which reinforces all the other screening parameters. And also some other potential applications would be, once again, considering the similarity between model organisms as entropy is a universal measure that can be applied in all kinds of model systems. A lot of applications. This is just a start, and hopefully we can continue. Thank you.
Questions from the crowd?
The main message, usually quite intuitive interpretation is that chaos increases with aging, right? But people, chaos. But that's an intuition. When you look at actual system and actual measurements, you see that actually the chaos is greater in healthy individuals, in young individuals. With aging, it becomes more regular, more normal, more routine. So that's the main message. This is a very formal. It was important for me to indicate that this is a very formal measure. It is important to have formal measures in the study of aging. So this is Shannon entropy, and that's the measure. The thing about such formal measures, we can talk from here to there about qualitative interpretations, what aging is, right? It can be accumulation of damage, it can be excess, it can be deficit, but actually information theoretical measures, they allow you a very formal definition, or can allow you a very formal definition what aging can mean decrease in time. Decrease of time.
Yeah, I'll tell you why. I'll let you in a minute. There is a theory called the cross-linking theory of aging, and it relates to extracellular matrix theory, because it means that collagen, elastin, all the extracellular proteins are cross-linked, and therefore they are less fragile. I mean, they're less flexible, and then if you measure their entropy, it will go down. So with age, Our extracellular matrix becomes rigid, it doesn't move, so the entropy is going down. So if you measure that kind of a component of our biological system, you see entropy down with age. So we have a straightforward example how entropy can go down with age.
It's everywhere. It's also in heart rhythm. It's in brevet. For example, the Parkinsonian tremors are characterized by very clear regularization of the pattern. So the entropy once again goes down. The system becomes more regular.
But how can you explain all the combination of mutation, for instance, aging, perturbation, circulation, all the genetic effects that are increasing age, which brings up the chaos, like he said, because the entropy, this is one thing. The second thing, I have a little problem with the database that we use. PDN, the idea of the PDN is that they have much higher prevalence of diabetes. And the reason they have this high prevalence is because the environment changed. They started to consume Western diet, and that's what they caused. So I think there is bias with assessing entropy in this population.
Absolutely. This is just, as I mentioned, this is just a pilot and just the easiest database that we can get. We hope to expand it, and by the way, if anyone has available data sets with clinical materials in relation to age, that would be fantastic. The more data, the better. Of course, it's just a pilot, but this kind of finding agree with all the others, with the Goldberger's finding about the heart rate and brain waves, and I will mention the molecular changes. It all indicate the same direction. Yes, I agree that there are certain appearance of chaos with aging, certain mutation, but it is an intuitive interpretation. When we talk in formal terms like Shannon entropy, we see that the entropy increases. But another point that can be that, for example, is in a certain system the entropy can decrease at the expense of all the others. So, for example, in this particular set of parameters, we can show a set of entropy which can be at the expense of increase of entropy in others. But once again,
but the question is, with aging people die, so pluralism is also decrease. So do you assess the entropy individually or globally?
We always have to consider entropy with reference to a particular set of parameters. So if we took this set, we will find this kind of entropy changes. But it is very difficult to generalize. Once again, then the interpretation become informal and intuitive. We can tell.
Thank you very much. We are going to have two presentations by the students that won the prizes today.